So, welcome everybody to the Symposium Assessing Brain Connectivity from EEG and MEG Measurement that we are going to have for the 2020 World Congress of Psychophysiology, IOP 2021. In this presentation, we will have 4 speakers: Dr. Sepideh Sadagiani from the University of Illinois, Spatial and Temporal Principle of Electrophysiological Connectome Dynamics; Dr. Alexandra Aisolin, Martino Center for Biomedical Imaging, Patient Clinician Brain Connectivity during Pain Treatment and EEG Hyperscanning Approach; Dr. Guido Nolte from the University of Hamburg, relation between linear and nonlinear coupling measures for Gaussian distributed data; Dr. Flicker Hendricks from the University of Amsterdam, the relation between the phase lag index and lag coherence for assessing interactions in the EEG-MEG data. We first want to thank all the speakers for taking the time for participating in this session, all the time that they have taken for preparing the presentations, and we will go directly to the first presenter, Dr. Sepideh Sadagiani, who is going to give an introduction of this session and also present her work.
Hi, everyone. Thank you, Jorge, for the invitation and introduction, and thanks for all the organizers and those of you who are joining us to listen in. So I will be talking about large-scale connectivity, as I'm interested in whole brain connectomes. I will cover... Yep. Sorry for interrupting you, but maybe you want to know that it's not in presentation mode. Oh, I'm sorry about that.
Okay. Well, it's very visible no matter what, but just if you want to make it bigger. Perfect.
I'm sorry about that. All right, let's restart. So large-scale connectivity is the focus of my work and my talk. And although I will be really focusing on electrophysiological connectomes and scalp EEG, as well as intracranial EEG, I will have throughout the talk comparisons to fMRI connectomes, because that's really the modality that has been driving, historically speaking, the connectomics field. So I hope that this talk will really convince you that electrophysiology can contribute a lot to studying large-scale connectivity as well, and in fact informs us far beyond fMRI. I'll start by a section on so-called time-averaged or static connectivity. First looking at the spatial convergence with fMRI-based connectomes that are well known, as well as convergence between different connectivity measures within electrophysiology, and then finally the stability of electrophysiological connectomes across different mental states. The second part of the talk focuses on time-varying connectivity, so dynamics. And there I will be again focusing on the degree to which these connectome states or dynamic reconfigurations of connectivity patterns converge or diverge with respect to fMRI. And then finally, I'll show some principles in terms of canonical neurocognitive network patterns that can explain connectome dynamics in electrophysiology. So let's start by the static connectome. So again, with an interest in large-scale connectivity, I just wanted to give an overview to those listeners who are not very familiar with how that might be done in electrophysiological data, such as MEG and EEG. Typically, you would start with your sensor level pre-processed cleaned up signals. You might choose to filter them in canonical bands, for example, source localize them to a mesh, and then average the signals according to your sources, for example, choosing a functional or anatomically defined parcellation, as we typically would do in neuroimaging. And then you can focus on the phase time series or the amplitude envelopes. look at temporal relationships between each pair of regions within any given frequency of interest. And you can do that, for example, for phase or for amplitude. You have to take great precaution here in terms of, depending on your interest, in terms of avoiding impact from source leakage, such as by approaches disregarding, for example, the zero lag phase relationships, or orthogonalizing signals between the different sources, for example. You can obtain a whole brain connectome, all regions by all regions, and here the color scheme is just the strength of connectivity based on phase or amplitude, for example. Once you have a connectome, you can of course move to topological space, use graph measures, or do anything else that you would be familiar with from, let's say, the fMRI or structural connectomics field. One reason that whole brain connectomes in electrophysiological data are not as prevalent as in fMRI is the technical difficulties with respect to source leakage and other considerations, as I alluded to earlier. But another aspect might just be the large amount of information, the complexity of electrophysiological signals. One aspect of that is that connectivity can be derived in various different ways. Here are two common ways of conceptualizing connectivity, for example, with respect to amplitude or phase, as I mentioned earlier. So if you imagine two different brain regions with time series here in red and blue, these are band-specific filtered time series, they could be locked to each other with respect to their phase, or with respect to their amplitude, or both. So in addition to that, electrophysiological connectivity may span the infraslow range, which is typically what we would look at in fMRI, all the way to 4 orders of magnitude above. So it's just a really, really rich signal that can be intimidating, but that can also really allow us to understand the brain at the time scales and with the mechanisms that it really is operating at. Just some older literature to convincingly showcase that large-scale connectivity can be observed in SCALP, EEG, and MEG. This is an MEG data set with seed-based connectivity in a broadband signal, looking at amplitude coupling, and a variety of these canonical large-scale networks can be identified as we know them from fMRI. As I showed you earlier, though, we can also generate the full connectivity matrix. So what I explained earlier was the source signal can be essentially estimated for a given parcellation, generating these connectomes for any given EEG frequency band. And a similar approach is taken for fMRI. This is something you probably are all familiar with. If we're using a similar atlas, we can actually correlate the connectivity strength of all the edges. So each dot here is one edge or one region pair. And compare them between EEG and fMRI. In this case, we're looking at imaginary coherence in the beta band as a function of fMRI connectivity strength. And you see a pretty convincing, although small to medium strong connectivity value here. What I find astonishing is that when you look at the relationship between functional connectivity, either for EEG or for fMRI, in relation to structural connectivity as estimated from tractography, you see a very strong relationship for EEG as well. So here it's just negative because of the particular DTI measure that was used. But what's interesting here is the strength of connectivity that for EEG is really strong compared to what we see in terms of fMRI to structural relationship. So this should really convince those who haven't tried EEG or MEG-based connectomes yet in terms of the ability to estimate sufficiently reliably large-scale connectomes. This is another study by my former postdoc, Jonathan Bierzig. He has estimated this same correlation value between edgewise connectivity strength and EEG and fMRI for various different EEG frequency bands. And interestingly, comparing 1.5 Tesla all the way through 7 Tesla, 64 channels to 256 channels in the EEG and various different healthy subject populations, you see that this connectivity is convincingly observed all the time, but also that the effect size is always moderate. It never reaches really a much larger value, and that's irrespective of the number of subjects that are included. So here you see that we reach something of an asymptote rather quickly, actually shockingly early, and adding more subject doesn't really improve on this spatial relationship between the two modalities. Of course, there is an unlimited number of reasons for why the relationship wouldn't be stronger, but I want to suggest that there is actually a neuronal difference between the activity or the neural populations that can be well observed with EEG MEG versus what fMRI can observe. So I'll get back to that towards the end of the talk again. Here, just to show an intracranial data set by Danny Bassett's group, showing a very similar effect size in terms of spatial correlation between intracranial connectome and fMRI-based connectome. Here, the edges are merged over subjects, because obviously every patient has only partial coverage. But what I want to point out here is the reliability of this relationship in the absence of source leakage, as well as this very, very similar effect size. I'm jumping through the rest of the talk a little bit back and forth between intracranial and scalp EEG, so I'm going to denote this with a red eye for you to observe it more easily. All right, so this is another intracranial data set from a study in our lab. I'm showing 4 example subjects here, and the subjects have different electrode coverage, so you cannot really compare across the rows. But what I want you to do is compare across the columns here for amplitude coupling and a measure of phase coupling. What you can see already just visually is that there's quite a strong spatial similarity between the different connectivity measures. And when we actually quantify that, that's the black data that I'm showing here for the different frequency bands. The spatial correlation between the connectivity matrix in phase coupling and amplitude coupling is fairly strong in all frequency bands, even though, again, it's not really going beyond a particular effect size here. So the individual dots here are individual patients. Another point, though, that I want to draw your attention to is that it really doesn't matter whether we look at the spatial similarity between phase and amplitude coupling after stimulus presentation, so during an evoked brain state, versus prior to stimulus presentation, which is more of an intrinsic brain state. So we tried this for various different tasks, including simple motor tapping, finger tapping versus more complex verb generation. In any different task, we always see the same pattern where looking at the similarity between phase and amplitude coupling prior to the stimulus, shown here in gray, really traces the same strength as the post-stimulus comparison. So much so that subtracting the pre-stimulus similarity from the post-stimulus really accounts for all of the data. So this points again to the fact that even in electrophysiology, there is a connectivity organization that is present irrespective of evoked state. So it's intrinsic to the brain. And a final aspect here in the static section that I want to point out is this mental state invariance of intrinsic connectome organization. So this is a connectivity matrix in fMRI for various different cognitive tasks compared to task-free resting state. And there is an astonishingly large spatial correlation between these two. This is an effect that has been replicated numerous times in fMRI. We've shown that this is also the case in intracranial EEG. So this is for one particular patient, phase connectivity, at resting state and during a cognitively demanding 2-back task. And you see a relatively strong spatial correlation between these two. So this is for the theta band, and we saw a very strong relationship as well in the beta band. We have quantified this for various different patients and various different cognitive conditions. We always see a fairly convincing relationship. In fact, the similarity between task and rest, or between different tasks, is no weaker than between retest of resting state. So there is an intrinsic organization that really doesn't change much between the different mental states. And so this is the summary up until this point. The functional connectome can be meaningfully studied using skull and intracranial electrophysiology. The electrophysiological connectome is shared across frequency bands and coupling modes. And connectome organization is highly stable over mental states in electrophysiological data, similar to what we know in fMRI. So let me take you to the dynamic connectivity section. I'll present two studies here. But first, let me motivate for why we should be looking at connectome dynamics. So this is an opinion paper by Gustavo Deco and colleagues. And so what you're seeing here is just a cartoon of a static connectome. So this would be similar to what I've presented up until this point, where connectivity is measured or averaged across the entire recording period. But if you break down the time window for which you're estimating connectivity, you'll actually see that this connectome organization, although it's the essentially average of these, really varies from one time point to the other. And these are typically called connectome states. And why we should care about it is that these may actually be of cognitive significance. So resting state dynamics, so intrinsic connectivity dynamics, is the gateway to the array of cognitive architectures that the brain has available. In a sense, resting state networks represent explorations by the brain of what is possible. If you're interested, we can discuss later on some evidence that I have in terms of how these switches actually impact cognition. For now, I'm going back to resting state data. And here, I want to draw your attention to this very rare data set where we have intracranial EEG concurrently with fMRI. The data is generously shared with us by collaborators from UCL, including Louis Lemieux's group. So big thanks to that. So what we're doing here is estimating the connectivity matrix in a given patient for fMRI and in the simultaneously recorded EEG as amplitude coupling or as phase coupling. Here I'm showing the spatial correlation between fMRI and phase coupling as a function of time. And you see that there is quite a bit of change over time here. And so, again, we quantified this window by window connectivity similarity between EEG and fMRI. And then we're thresholding it according to a phase randomization null model. So what I'm showing you here in the different colors are time series for the different EEG frequency bands in terms of their spatial correlation to fMRI. The colored blocks are marking the time points for which in the given frequency the similarity surpassed this threshold. One observation that's interesting here is that fMRI becomes spatially similar to EEG for the different frequencies at different non-overlapping times. So it's not an EEG broadband phenomenon that happens across the connectome, and that's then metabolically translated into the fMRI signal. So one question here is fMRI, if we take this color analogy, is fMRI something like a black and white goggle that cumulatively merges over everything that that EEG can see in terms of this very broad and detailed frequency-specific connectome dynamics phenomenon? I argue that that's simplistic. That is not necessarily the case. One interesting observation here that speaks to that point is that over all frequency bands, if we sum up all the time windows in which we found significant similarity to fMRI, we only get to about 20% here over the different patients. And I'm not showing the data here, but we replicated this very sparse relationship and frequency-specific relationship in SCULP EEG fMRI data of healthy subjects. So we went ahead and did something different here. So this relationship is measured between EEG and fMRI at the same time. At any given moment, we compare what's happening in EEG and what's happening in fMRI in that moment. But if we actually slide them across time, we get a repetition plot or spatially shifted relationship between EEG and fMRI. So time is running on both axes. And at each data point, we're comparing the spatial correlation, comparing EEG and fMRI in terms of spatial correlation in a time-shifted manner. So along the diagonal would be synchronously occurring connectome states. And what was shocking to us, and again, we replicated this in SCOP data, is that the ratio of time points showing significant correlation at the same moment in time as well, contrasted with shifted time points, the ratio is the same. So there's nothing special about this diagonal. EEG and fMRI show spatially similar patterns, but not due to the same neural phenomena because they're not synchronized in time. They're spatially asynchronous. And we have another study, and I will stop right after this, that really convincingly gives us an understanding for why this asynchronous aspect would actually occur. So what we did here in SCULP, EEG fMRI of healthy participants at resting state, is we're looking at spatial binary maps. This was introduced recently by Olaf Sporns for fMRI. And these maps, each one of them, which is one column here, just is ones versus zeros for one of the canonical Thomas Yeo networks, so 7 canonical neurocognitive networks here that can either be individually activated or as a pair or as triplets and so on. We have the full basis set of 126 states. And what's interesting is if you map for concurrently recorded EEG and fMRI at every time frame, the best fitting template, and you assign it to the time series. So for this time point, I would look which one of these templates best explains my instantaneous coactivation pattern. So I choose that and I move on. If you do that for every EEG frequency band and for fMRI, you see that the same spatial templates are present in every frequency band of EEG and in fMRI, but they're stacked in a temporally independent manner, such that faster and faster time scales are actually showing faster and faster switches across the same spatial coactivation patterns. And so this is the overall correlation of how well the best assigned templates are fitting our data, and this is far significantly above chance level in a null model. So this holds true for fMRI as well as all EEG bands. And so the way we're interpreting this is that actually the connectome is switching between different connectome states, which are explained by these network coactivation patterns. And if you zoom in into a certain area of this and look at a finer and finer time scale, you see a scale-free pattern of the same transitions across the same templates. And in fact, when we look at the transition probability matrices that I'm not showing here, they're spatially incredibly similar. So even the sequences taken and the probabilities of transitions between these states really match well across the different time scales. And what that indicates to us is that hemodynamic connectome dynamics and electrophysiological connectome dynamics may be largely non-overlapping. So these are different neural processes on a scale-free continuum. These are not synchronized in time, even though they share the same spatial information. So let me close here, summarizing. There is robust cross-modal similarity of static connectivity. The spatial similarity of dynamic connectome states occurs asynchronously across electrophysiology and fMRI. And we have a connectome dynamic scenario that is temporally scale-free, covering all speeds from infraslow in fMRI all the way up to the gamma band. So we really, to understand connectivity, need to look at the full spectrum of what the brain has available. I'd like to thank my lab, especially Tom Haram and Jonathan, from which I showed the data today. And thanks a lot for listening.
Thank you very much, Cepides, for very nice presentation. Let's go directly to Alexandra's presentation to see if we have time to make some discussion at the end.
All right. Can you hear me well, and can you see my presentation?
Yes, perfect.
Perfect. So first of all, thank you so much for having me at this conference and this symposium. I am really happy and very curious and inspired, from the topic that the other speaker on this symposium are gonna touch. And I loved Cepides' presentation. And that actually helps me a lot, because here we're going to talk about another layer to add to EEG analysis that is including data from two subjects at the same time, starting from the knowledge we already have of this pipeline that she walked us through. So today I will present the study where we recorded the brain activity using a simultaneous EEG from interacting patients and clinicians to assess the effect of therapeutic alliance in a specific clinical context, which is the one of the pain treatment. And of course, it's tough talking, touch the topic of social science and social interaction during COVID, but we came up with a solution that involves these clear masks. I don't know if you can see my mouse, which allowed us to keep carrying on the project even during COVID. Okay, so we are interested in understanding more about the brain mechanisms underlying therapeutic alliance and the role of therapeutic alliance in chronic pain therapy. It is a relatively new field of research in the neuroscientific panorama, because for years it's been considered kind of an intangible art of medicine. But the reality is that the patient-clinician relationship is foundational to healthcare, and that plays a key role in several aspects. This starts from the patient satisfaction, going through the clinician satisfaction, quality of life, and most importantly, clinical outcomes. For example, the paper I cited in this slide demonstrated significant changes in the clinical outcome in pathologies such as depression and chronic pain. And just to give you an idea, a quantitative idea of this effect, I reported here the results from one of those papers in which patients with irritable bowel syndromes who had an augmented intake with their therapist reported an improvement in symptom severity, which is double compared to the group who had a limited interaction with their clinician. So patient-clinician interaction can powerfully shape treatment outcomes such as pain. And what is pain? Pain is something that is stereotypically associated with the tissue damage, but actually is much more nuanced experience, is very subjective, and is influenced by factors that play a role in such experience. And among these factors, we have for sure biological factors. For example, if some fibers are exposed too frequently to painful stimulations, they might start amplifying the signal they send to the brain, causing sometimes a condition that is called the chronic pain. There are for sure psychological factors, which are important because they can affect both the nociception system and the brain directly, and social factors. And this is the dimension in which therapeutic alliance can shape pain perception and produce analgesia. So the role of psychological factors has been studied several times, but for sure there is still so much to understand about the brain mechanisms during something complex and unpredictable, such as the social interaction. So brain correlates of social processes such as empathy, theory of mind. I've been studying using single subject designs, but specific behavioral and neural mechanisms underpinning the patient-clinician interaction are still unknown for this reason of not having a full picture of the design. So most of the time, the patient's response only was studied with a few exceptions. And so one of the reasons why in the past studying social interactions was challenging is that very unique processes happening when two people interact that cannot be fully explained by studying individuals in isolation in a lab. And so it requires what we call two-person neuroscience, which is fundamentally based on two things. the simultaneous brain activity acquisition from all the interacting subjects produced a procedure called hyperscanning, and multi-subject analysis, which is as important as the simultaneous acquisition, because if you are not able to look at multiple subjects' data within the same model, we might still lose important information and the brain responses. So let's start talking briefly about the acquisition. So EEG hyperscanning is advantageous for multiple reasons. The high temporal resolution allows for a more dynamic investigation of the brain during social interaction, and the kind of a flexibility in analyzing that unpredictable part of every social interaction. And speaking of naturalistic interaction, for sure, EEG allows like face-to-face communication, which creates a more ecologically valid experimental design, which is missing, for example, in fMRI hyperscanning design. And is very important because a big part of social interactions is nonverbal communication. Additionally, EEG hyperscanning opens the floor for multi-person studies with N greater than 2, and there are already in literature several like cool examples, as for example, like alumni and teacher in a classroom, or a quartet of musicians playing on stage. Of course, all of this doesn't come without some challenges, including the necessity of a perfect synchronization of 2 or more systems, and the importance of either recreating in a lab in a very naturalistic environment, or employing like portable and Wi-Fi EEG systems. In terms of multi-subject analysis, there is a lot to discuss in terms of dealing with the time series coming from 2 brains at the same time. But I will keep the focus on the concept of brain-to-brain connectivity. So here we can see how the data extracted from subject A and subject B are concatenated across the channels. So if, for example, we choose a standard montage with 64 electrodes, our omnibus matrix will contain 128 time series for each epoch, which might be, for some analysis, pretty expensive in terms of computational time, especially if you're talking about multivariate autoregressive models. A common practice to deal with this are selecting a subset of electrodes on the scalp, or doing source reconstruction, and like as Silvia was saying before, either going through a parcellation or like choosing a specific inadequate number of ROIs. Anyway, after reducing this dimension and an appropriate normalization of the data, a multivariate autoregressive model can be run, considering all the time series at the same time, and unveiling in this way causal links and relationship associated with the information flow between the 2 subjects, clearly hidden if we analyze only the activity of one subject. So the combination of hyperscanning and multi-subject analysis has been already successful, used in several interesting applications, mostly on healthy subject, I would say. So here we have juggling, games, pilots during a flight simulation, musicians 2 or 4 or 3, joint actions, and nonverbal communication between a parent and a child. But like our lab in Boston is the pioneer in the use of hyperscanning techniques for investigating the relationship between patients and clinicians. Before the current EEG design, which I'm presenting today, another project, like another hyperscanning fMRI study, was performed on fibromyalgia patients and acupuncturists in order to like shade a light on the social mirroring, synchrony, and brain concordance during this interaction. So our hypothesis was and is that patient-clinician interaction is fundamentally based on mutual empathy, therapeutic alliance, behavioral mirroring, and synchrony, for example, in language style or gesture. And on top of this, of course, concordance, synchrony, or communication between the 2 brains. The first paper out on this topic was published in 2020 by Dan Michael Ellinson, former postdoc in my lab. And it actually shows, among other things, the therapeutic alliance and brain concordance, specifically in a region called the temporoparietal junction, are enhanced by patient-clinician interaction. And also that the greater alliance is linked with enhanced facial mirroring. So this paper provided very important indications of the neural mechanisms for social interaction, but it comes with some limitation because we know that the 2 people, like the patient and the clinician, were lying into scanners during the actual interaction. And so this is where the EEG understanding project enters the field, maximizing the ecological validity of the experiment and looking beyond what we call the brain concordance to look at causal relationships between patients and clinicians' brain. So let me dig very quickly into the experimental design of the study. So despite COVID, we recorded 24 diets so far, dividing into groups depending on whether the clinician was instructed to perform an augmented or limited clinical intake. So we have chronic low back pain patients on one side and the MGH licensed acupuncturist on the other. And the very first part of the experiment, of course, is the EEG setup. So we used the 2 EGI system with 64 water-based channels net for each subject. Plus, we acquired the cardiac data, so ECG, skin response, and facial expression, everything synchronized with the EEG systems. So hopefully we will try to include all these aspects in the model. So the first acquisition is the most naturalistic one. We call it intake. So here the clinicians complete an augmented or limited intake based on the diet randomization. And so if they are augmented, it allows for a long personal conversation about patients' experience with their pain. It involves an active listening, personalized content, while if they are limited, they run through a quick intake checklist with the patient that is more neutral and narrowly focused on specifically and only the pain. Of main interest for this presentation is the pain task that follows the intake, in which patients experience evoked pain delivered by inflating a cuff around the lower left leg. Importantly, we defined like 2 experimental conditions that we'll call treatment and no treatment, depending on whether the clinician does or does not treat the cuff pain using an electroacupuncture device. And the painful stimulation lasts like is 10 seconds long and gets repeated like for 20 times. So after each cuff pressure, which is calibrated in order to evoke the moderate pain, the patient rates the intensity of the pain they felt with a pain rating scale, while the clinician rates patients' vicarious pain. So to summarize, we have 2 important factors that we will bring in our statistical analysis, which are like a within factor, which is the presence or not of the treatment, and a between factor, which is the clinical context that can be augmented or limited. So after the interaction with the clinician, patients fill out a number of questionnaires, including the care and the warmth and competency scale. The care is used to measure the quality of the patient-clinician relationship rated by themselves, like separating the patient and the clinician, while the warmth and competence scales is used to measure patients' subjective ratings of clinicians' level of warmth and empathy versus like their level of competence. So here we can see how care scores and warmth scores are significantly higher in the augmented group than the limited, with no significant differences for the competence. And this serves as like a nice check that our augmented group is successful in creating a warm and empathetic bond with the patient, where the limited group is clearly not. And however, also the lack of significant difference in competence is a good result, as we still want to successfully recreate a clinical setting in which sometimes clinicians may spend less time and build less of a bond with their patients, but still be competent doctors. As a clinical outcome, clinical low back pain intensity was assessed before and after acupuncture, and we found in both groups that back pain was significantly decreased after acupuncture. And additionally, we measured the difference in the pain rating during treatment and no treatment blocks of our experiment, and found that while the pressure stayed the same for both conditions, pain was significantly reduced during the treatment for both groups. And interestingly, we found that the clinician perception of patients' pain and analgesia during treatment was significantly correlated with the patient's self-rated analgesia, suggesting that actually clinicians can perceive via no-verbal communication the pain relief that the patients experience during the treatment. So before describing the connectivity results, I want to go back for a second to the methods employed in this analysis. Like it is pretty like a standard pipeline that we already saw in the previous presentation. So we started with the pre-processing, just to highlight that we bandpass our data in the between 1 and 45 hertz. And the independent component analysis was applied to identify and eliminate ocular movements artifact, which sometimes not really during the pain task, but during the intake, for example, are particularly high when there is like an active speaking and communication between the 2 subjects. And as in every upper scanning design, common trial rejection was mandatory, reducing the amount of usable data of more or less 30% on average. The second step is the brain source reconstruction that was performed employing the algorithm Exact-Loretta because of its high performances in avoiding introducing error in the following connectivity analysis. We selected 17 ROIs for each subject involving brain regions of the social mirroring, empathy, and brain processing networks. And conveniently, the coordinates of those dipoles were extracted from our fMRI upper scanning paper, targeting the same patient-clinician interaction. Source reconstruction is followed by the M-bar coefficient estimation and multi-subject partial directed clearance matrix computation, where this time we obtained this omnibus matrix, which is divided in these 4 parts coded by colors. And here the red codes for the network estimated within the nodes of the patient, purple within the nodes of the clinician, blue in the top right corner represents the link directed from the patient to the clinician, and the light blue, the opposite direction from the clinician to the patient. And lastly, we quantified some of the properties of these interbrain networks. So in the previous example, the blue and light blue part of the matrix, resorting to well-known graph theory indices that allowed us to perform also some statistic analysis using those measures as independent variable in a two-way ANOVA design with the treatment and the treatment as the level of the within factor and augmented and limited levels of the between factor. Specifically, we computed interbrain density, interbrain strength that considers also the intensity of the connections rather than their existence and directions only. And additionally, in order to identify meaningful nodes with a key role in the information exchange from one brain to the other, we also computed the same measures at single node, or in this case, single ROI level. So focusing here on interbrain connectivity, the main novel results we found is that the number of connections between social mirroring and nociception processing regions is directionally modulated by the presence of the treatment during the painful stimulation. So from clinician to patients, we see a higher number of interbrain connections during the no treatment condition, suggesting that social mirroring and empathy are stronger on the clinicians when they experience vicarious pain of the patients in pain, and when they basically cannot even treat them. And on the opposite direction, the number of interbrain connections from the patient to the clinician is higher in the treatment condition, suggesting higher social mirroring when patients know that the clinician is actively treating their pain. And importantly, those results are true only in the augmented group, because if we look at the same results for the limited group, we can see that we cannot see the same behavior. Here, what we can see is, in general, less interbrain connections from the clinician to the patient and vice versa. And in general, also higher interbrain density during the treatment, regardless of the clinical context, so confirming that there is certainly more social engagement with respect of the no treatment, even without like personal bonding. So here, this plot offers just a statistical quantification of the results we just commented on. So both density of interbrain networks and the strength of the links are higher in the no treatment versus treatment only in the augmented group, as we can see here, confirming the directional modulation of the brain-to-brain interaction. And here we were focusing only on the clinician-patient direction. While if we focus on the patient-clinician direction, same thing, we can see how for both augmented and limited group, there is a noticeably higher number of interbrain connections, but the strength of those links is significantly higher during pain treatment only in the augmented group. And finally, like about involvement of specific brain regions in this brain-to-brain information flow, we found very interesting results. This is the way in which we can actually look at a parallelism with the fMRI results, because those results are supported by previous evidences derived from fMRI studies about the same brain areas during empathy for pain, pain processing, and social mirroring. And on this regard, we decided to collapse augmented and limited groups and actually focus on better characterizing the brain-to-brain response during pain treatment versus observation of pain. So first, we can notice how in the no treatment, more than in the treatment, there is an impressive overlap in the way maps, like especially here in the right corner. So this is like, we can see, for example, the same dominance in the sensory motor areas and the same modulation of the engagement of all the other nodes. Second, to give a taste of the main findings related with previous evidences, I want to mention above everything like a TPJ, or the temporal parietal junction, which might be one of the more robust results we found. Specifically, the degree of TPJ, so we are looking at the first line here, shows that it is one of the dominant nodes connecting patients and clinician brain activity, which is a very relevant considering that TPJ was also the main region showing brain concordance in our previous patient-clinician fMRI study. Interesting is the fact that left TPJ is dominant in the patient during the pain treatment, and in the clinician, we have right TPJ during the no treatment, where it is well known that right TPJ plays a role in the way individuals observe and process information, impacting social interaction, and this makes total sense considering that the clinician in this phase is clearly like observing the patient in pain. Under a more social science lens, it might be also an important additional information to the model we hypothesize, in which there is a prevalent direction of edges in the interbrain network, depending on the role of the clinician during the trial. So there is a difference when the clinician is the person who gives relief versus when the clinician is the person who observes and feels the patient in pain. So just to summarize very quickly, we understand how important it is in order to answer to the question, are brain mechanisms across the patient-clinician diet significantly affected by the interaction style during pain treatment? We really need a two-person neuroscience and to include both subjects in our model. So we need this combination of hyperscanning and multi-brain analysis tools. From a more brain perspective, we saw how during the augmented interaction only, the density of the interbrain network directionally modulates with the presence of the treatment in a way that is very specific for the patient and the clinician. That during evoked pain, a more intense exchange of information connections during treatment suggest a higher engagement of the clinical diet, regardless of the context and then the bonding, and that involvement of specific brain regions within the interbrain network reflected their physiological role and confirms our previous fMRI finding. So the very last thing I wanted to share with you are like the idea for the next analysis I have. Of course, there are so many limitations associated with the fact that it is a new field, and there is a lot to explore for sure. Methodological advancements for hyperscanning data analysis and visualization and validation needs to be done. So I recently published like a paper in which I presented a toolbox for the generation of simulated data for testing the connectivity algorithms. Like we are trying to extend the generation of simulated data also to the multi-subject case. So hopefully that will help to understand the accuracy of our results. And for sure would be important to include like other connectivity measures and other autonomic signals. And the one important part of this from a more psychological side is to include the preference of the patients, because the more I interact with them, the more I understand that augmented and limited might not mean the same thing for everyone. So we included this questionnaire that is called COPRA, developed by a German scientist, and that is helping us to keep track of the personal preference of the patients. So I would like to thank my current lab, like the Martino Center, my former lab in the Sapienza University of Rome, and all my mentors, Vitali, Ted, Laura, Ilenia. And thank you all for your attention. And I really am looking forward to have a discussion during this conference.
Thank you, Alexandra. Very nice talk. This is also another passionate work. Let's go to Guido's presentation.
Is now fine from...
Yes,
Yeah,
perfect.
okay.
Yeah.
Okay, now it's good. So I will talk about relations between linear and nonlinear coupling measures for Gaussian distributed data. This is just one work. It's not completely new, but it's relatively new, like 1 year old. And I think when I noticed, I was happy to see that it's probably relevant for both previous talks. At least there are some aspects about it, and surely also the last talk we hear here. So there are different ways we can look at coupling between two signals at two sites in the brain or in sensors in the frequency domain. The most typical ones are phase-phase coupling and amplitude-amplitude coupling. So on the left here, you see phase-phase coupling. That means in this example that the peaks are always at the same time point. They're always aligned, but how strong the signal is, this is completely independent between the red and the blue. So they are phase-phase coupled. What you could also have is an amplitude-amplitude coupling. Here, the peaks of these oscillations have nothing to do with each other, but the amplitude does. So when one is large, the other is also large. And this looks like two completely independent phenomena, which could be used by the brain for some kind of communication with different modalities. But the question here is whether these phenomena are really so independent as they look when they look like when you just look at these figures. Actually, all we do here all I do here is talk about coupling within one frequency. It's not a cross frequency. That's a different thing. And this is a completely nonlinear phenomenon I will not talk about. So basically, to study phase-phase coupling, there are two main methods, I would say. One is coherence, that's a linear measure, and the other one is phase lock. That's actually a nonlinear measure. And to study amplitude-amplitude coupling, the typical one is we calculate amplitudes by Hilbert transform, and then, for example, we square it or not and look at the correlation between these two signals. So that's then the question, are these phenomena independent of each other? First of all, I will talk a lot about linear measures. I make the relations between linear and nonlinear measures for Gaussian distributed data. So let me first recall what linear measures are. There's only really, if you do bivariate analysis, there's really only one thing, and that's a cross spectrum, like the variance you all know between two sensors, say I and J. So this is just using data in the Fourier domain. Everything becomes complex in this second order moment. This is then the cross spectrum. And this cross spectrum you can normalize by the diagonal. That's the power, in fact, and then you can have something like coherence, or in the complex way it's coherency. It's a complex number. The absolute value tells you how strong it is between 0 and 1, and the phase, whether the peaks have a phase delay. Okay, very important here is a function calculated from this complex coherency, and that's lag coherence. It's still a linear measure because it's just a function of this linear term. It's just re-expressing it. So it's lag coherence is the imaginary part of coherence divided by square root of 1 minus real part squared. So it's just a function, and we will see it becomes relatively important here. Now, the linear measures have a specific advantage. So if you decide not to use a linear measure, you should have a good reason. And I would go mention here some advantages. So usually you can calculate these linear measures in sensor space and then transform results into source space easily. This is what you do when you have a linear inverse method, which is very common. Like you could do it with beamformer, also with E-Loretta, what we've seen in previous talks, for example. Second, it is relative. you have relatively simple analytical calculations if you want to maximize something or so with these matrices, cross-spectrum matrices. So it's not very difficult to do. And finally, and that's very also quite important, you have additivity, cross-spectrum of independent sources. And this becomes also relevant when you want to correct for artifacts of volume conduction, for example. So these are the advantages. One could add that it's typically a very robust measures, and often nonlinear measures are not so robust. So there are good reasons to do it, but you might have other reasons not to do it. So, and the question is when. So the point here I want to make is, if you have a linear dynamical system, data are Gaussian distributed, then there's no real reason to do to use a nonlinear measure. So let's talk a little bit about Gaussian distributed data. What you have here, this is one example, empirical example of filtered data. You can see this here. And when you look at the distribution of this data, you find it's perfectly Gaussian distributed. This is an extreme example. It's not so perfect usually, but usually it's very well Gaussian distributed. So often you can make assumptions that it is Gaussian distributed, which, however, can also be wrong. Anyway, so assuming that data are Gaussian distributed is usually not so bad. Sometimes deviations are relevant. Second, you might have a model. So you have your data and you want to fit some model and interpret it. That's what you do. And so we have also seen this in previous, say you do PDC. What do you do then? You have your data, you fit a linear autoregressive model, and then you interpret the parameters. But then basically, if you do that, you assume that your that linear dynamics is valid. So basically, you assume then also that your data are Gaussian distributed. Finally, why it is important, because sometimes you interpret your results in a specific way. And to make this interpretation right, you must assume it's Gaussian distributed. Otherwise, your interpretation is not wrong. I will show you later an example of that. But first of all, this is why Gaussian distributed are relevant. But now, what are the consequences if it's Gaussian distributed? So I would say, you have a stationary linear process, then you have for the data in Fourier domain have a very specific probability density, and it's just a Gaussian function. So you have your data, say Z, in the frequency domain, inverse cross-spectrum matrix here with the adjoint times Z. That's it. So furthermore, for all we discuss here, the total signal strength, like the power, is irrelevant. So we might normalize it to have power 1. Then the entire probability distribution is given by one coherence matrix. So you have 1, 1 here, that's the power is 1 in the diagonal, and you have the complex coherency in the off diagonal. That's all there is. If you know complex coherency, you know the entire distribution of the data. And if you know the entire distribution of the data, you can calculate anything you want. You can calculate any statistical measure, any nonlinear coupled measure, just from this probability density, and then hence just from coherence. So there must be some functional relations. This is not really new. People know that, but the question is, what are these relations, and what can they teach us, and can we use that in any way? So this work is basically to calculate, to look at nonlinear measures, to assume there's a Gaussian distribution, and then to calculate such a relation mathematically. And this is what comes out. So the first example I show you, and to illustrate what this means, so this is a weighted phase lag index. Basically, the numerator is like imaginary part of the cross spectrum. Just the normalization is different. That has an advantage. It's a good idea, really. Anyway, but so this is a weighted phase lag, and it's very popular. And there's a specific relation to coherency. And basically, the relation is, this is a function of lack coherence. This is not identical to lack coherence, but it is a function of lack coherence as written here on the right. I want to illustrate what this means in simulation. So you have here, we make each point here is one dynamical system. You can say a linear dynamical system, and for that system, randomly selected system, and for that system, we calculate a linear measure and a nonlinear measure. So here on the left, you see imaginary coherence on the x-axis, weighted phase lag index on the y-axis. You see they are not so different, but there is not a strict relation between them. So weighted phase lag index is not a function of imaginary coherence, but it is, if you do it, we calculate where we show weighted phase lag index versus lag coherence, then you see there's a unique functional relation between them. But this is again, there's a functional relation between them, but it's not identical. But if we know what the functional relation is, then we just can calculate it, and then we see weighted phase lag index is just a specific function of lag coherence. And later we would consider this as a model. Say we to make a linear model for weighted phase lag index, we would calculate lag coherence and insert such a function. That would be called our model for it. Now this was just one example for the weighted phase lag index. This is now what you've seen before, but we have done it in fact for basically 5 or 6, depends how you count it, different measures. So another one is the phase lag index. And Ricard later in the last talk will talk largely about that. Phase lag index, there basically you take the average of the sign of the phase difference between two signals. That's the phase lag index. And it turns out that the phase lag index for Gaussian distributed data is identical to lag coherence. Again, note it's lag coherence becomes important here. Well, not for all measures. So here is 1 or 2, depending on how you count it. Phase locking itself, that's also a nonlinear measure. Phase locking is in fact, so as a complex number is equal for Gaussian distributed data equal to complex coherency with some scaling function. And this function makes it just a little bit smaller. That's all. And you can consider this as 2 measures because often you take either the absolute value or the imaginary part, which would be itself be considered as a measure robust to artifacts of volume conduction. Okay, there are 2 more. All these previous one were phase-phase coupling. Now it's amplitude-amplitude coupling. You can look at the correlation between powers, or also between amplitude. Power is just the square of the amplitudes, and it turns out that the correlation between powers is just the square of the absolute value of coherence. This has some implications. One is, if you have a linear dynamical system, you can never see a negative power correlation. So whenever you see a negative power correlation, it must be a nonlinear phenomenon. So finally, this power correlation, that's not robust to artifacts of volume conduction, meaning if you have just mixture in sensor space, you get confused. This could be, you just see a mixing artifact there. There are ways to address this and to make it robust to artifacts of volume conduction, so to say, and that uses that orthogonalizes this signal in such a way that if they are linearly related, the mixing artifact is removed. And it also turns out that this measure, I would call OPEC, is just the square of lag coherence. What you see here is that all these 3 measures, robust to artifacts of volume conduction, are just functions of lag coherence. One should add here, and this is really, I would say that's a problem in this case, this orthogonalized power envelope correlation is has the idea of being robust to artifacts of volume conduction, but it's only true if data are Gaussian distributed. This would be one of the examples where a specific way of interpreting such a coupling, you need to require that it is Gaussian distributed. Otherwise, that's in general not the case. So I'll show you here some simulation for that using a linear system, linear autoregressive model versus a highly nonlinear model, the Kuramoto model, which is just the dynamics of phases. Now I don't show the model themselves, just to show results. So each point here, again, is just one complete randomly selected linear system and with simulated data, and then we calculate both the nonlinear coupling and the model. Again, the model was we calculate the linear coupling, and from that we use a known function to say this is what we predict if the data are were Gaussian distributed. And if you have all linear models, you see here the blue dots is almost for all measures almost perfectly on the line. If we have if we have nonlinear dynamical system, and Kuramoto model is a highly nonlinear dynamical system, we see deviations, of course. These 4, 1, 2, 3, 4, they're all phase-phase coupling. Deviation is fairly small, so it doesn't really matter, you could say. But it becomes very relevant when you look at power-power coupling, both in this really just usual amplitude-amplitude coupling, or orthogonalized, which makes it robust to artifacts of volume conduction. Here is one empirical example. This is one subject we calculated for all channel pairs, linear measures and nonlinear measures, and compared them. So this is just one subject, but it's typical. You can see the same thing basically in all subjects, more or less all frequencies, but in principle it's very similar. So here, for example, is one thing to show, and this is very typical. If we have the model, that means a specific function of lag coherence, it's almost identical to the weighted phase lag index, which is, I can't see my mouse, which is on the upper left here. So there's not much different. And then you could say, if it's really almost identical, it really doesn't matter what you use. There's no advantage anymore. Results are similar for model and nonlinear measures for all phase-phase based couplings, PLI, phase locking, absolute value, imaginary power, but they are different for power or amplitude coupling. They can be quite similar usually for amplitude coupling if you do not orthogonalize, but there are substantial difference, and that's interesting to look at. And they are not just a little bit different, but they are massively different for this OPEC power coupling with orthogonalization, and there you see a huge difference. This huge difference here, they are not close to these lines, means that the difference you see must come from nonlinear properties of the data. They are not Gaussian distributed, even if minor. This makes a huge difference here, and this makes, in fact, a problem. If you've to interpret a OPEC, you assumed it's Gaussian distributed, otherwise interpretation is not right. And then you find, but you can check this, and then you find out this assumption is certainly highly violated, at least for this measure. And that makes it difficult to interpret that. So this was the empirical data. I want to show you one last empirical example from ferret data. This is I forgot what the kind of data we have, but there was as a function of frequency, we have some sensors in ferret brain, and we calculate coupling for all frequencies from 0 to 200 Hertz. And average across all sensor pairs, always with absolute value. Here, this is coherence. We average that, and we compare two conditions. One is a slow wave sleep, and the other is all the rest. That the slow wave sleep is so easy to detect, so we can compare slow wave sleep to everything else. And when we do this for coherence, we can find almost no difference at all. So coherence itself cannot distinguish the two states. But here's a way we can subtract. Subtract from power coupling the square of coherence, and that what we then see is a purely nonlinear coupling measure. All linear coupling was removed, and then we can see also clear differences between slow wave sleep and non-slow wave sleep, and this means that this difference is generated by nonlinear processes in the brain. So that was basically it. I want to conclude. So for linear systems, all coupling measures, they are equivalent to coherency. So if you assume that, you might just as well directly calculate coherency. There are statistical properties that differ, and I know that Richard will talk about that. In general, there are minor differences for phase-phase coupling. Amplitude coupling, that's the most interesting thing to me, because the differences are very large. If you also correct for artifacts of volume conduction, the differences are even higher. But probably that's less interesting, because it's more problematic than interesting. Because then what you probably see is then you just see artifacts of volume conduction. One measure has it, the other doesn't have it, and then you see huge differences, and that's not so interesting. So that's basically all I want to say. There's just one paper I have to say, so I want to thank you all my co-authors in that. So I've written it here: Edgar Galindo-Leon, some Chinese colleagues from Beijing, with whom we collaborate, and Andreas Engel. Okay, that was it. Thank you.
Thank you very much, Guido. Very important conclusions, very, very important study on the nature of these many very different connectivity measures that we are using. Let's go to Richard's presentation.
Guido, can you stop?
Yeah.
Yes, yeah. Thanks. So this is visible? Yeah. So George, thank you for inviting me, and thanks for the other interesting talks. And indeed, yeah, my talk has a lot of overlap with Guido's talk. But I will focus, and it's in fact inspired by Guido's paper. So I will focus on the phase lag index and the lag coherence. So in his work, Guido showed that these are in fact the same for Gaussian models, Gaussian data. So what that implies is that if you have two estimators, one for the phase lag index and one for the lag coherence, that they are in fact estimators for the same quantity, namely lag coherence. So, and then it makes sense to compare the sampling properties of these estimators and see which one is better. So that's what I will do in this presentation. First using simulated data and then empirical data. But let me give a short introduction for the people that didn't see Guido's talk. So the context is very simple. And I don't feel bad that I have equations, because Guido also had equations. So, okay, suppose we want to assess brain connectivity between two brain regions based on the reconstructed source signals. So, and the whole time we work in the frequency domain, as Guido said. So Z1 and Z2 are reconstructed signals. These are complex valued signals. But because of this incomplete mixing of the inverse operator, in general, your reconstructed time series are linear combinations of the true source time series. And in the simplest case, our two reconstructed signals are a linear combination of the true signals at the same locations. That's the most simple situation. So the least that we want from a connectivity measure is that it is invariant under this transformation. So if you have a connectivity measure F and we apply it to two source signals, reconstructed signals. So it means we apply it to two mixed signals. We want the same answer as that if you would have applied a measure to the two signals. So this transformation by this mixing matrix doesn't change the connectivity measure. So that's called an invariant measure, and that's what we want from these measures, to be sensible and interpretable. So, and two of these measures are the phase lag index and the lag coherence. So here they are, this is essentially the same definition as Guido gave. And I will focus on these two measures. So, and this goes about the interpretation of these, of the results of a connectivity analysis. It's usually, whether you use, so if you use lag coherence or phase lag index, the interpretation of the results is usually different. And the question is if that is really a good thing. So if you use PLI, then usually the results are interpreted in terms of synchronization between self-sustained oscillators. So in a non-dynamical way. But if you use lag coherence, the results are usually interpreted in terms of co-fluctuations or correlations between random processes. So these are dynamically very different. So for me, the important thing that of Guido's result is that for Gaussian signals, the lag coherence and the phase lag index are the same. So that means that there's only one interpretation, and that's the one. It has to be the same interpretation given for PLI and lag coherence. And in this case, you have to interpret it in terms of random processes, unless you can show that these two measures are also the same in a nonlinear model. So what we focus on is the following fact. So suppose we have two estimators, one of the PLI and one of the LC. Since they estimate the same quantity, namely this quantity, it makes sense to compare their performance, their sampling properties. So the most natural estimators are the following. So, and I indicate an estimator by a hat. So an estimator for the PLI would be, it's just obtained from the definition of the PLI, but I replace the expectation value by an averaging operator. So it's these brackets. And the most natural estimator for the lag coherence is obtained by replacing the rho by rho hat. And rho hat is just the sample coherence, which again is obtained from the coherence by replacing expectation values by averaging. So these are in fact the two estimators that are used in practice. So I want to compare their sampling properties, and in particular their standard error. So let's first do that for model. Yeah, so I have this Gaussian model, and we simulate a large number of signals from that, with different values of the coherence, and for different sample sizes from 1 to 200. And then for each combination, we compute the standard error. And what you see is very clear cut. For all values of the coherence and for all sample sizes, the standard error of the standard error of the PLI estimator is larger than that of the LC estimator. So it cannot be any more clear. So here you see on the x-axis the sample size. So that's the number of Fourier windows, or the number of trials. And on the y-axis you have the standard error. And yeah, they both scale as 1 over the sample size, so they go down. But the phase lag index is always larger than the lag coherence. So, the first conclusion is that if your data is Gaussian, then there's no reason, no justification for using the PLI over the LC, because it's always better in all conditions. That's the first conclusion. Okay, let's, yeah, the second thing I want to address is to see, yeah, if experimental EEG, MEG data actually come from this Gaussian model. And if not, can we characterize the deviations? And can we then also, if the data turn out to deviate from that model, can we then, is it then still true that the lag coherence is equal to the PLI? And is it also still the case that these estimators perform in the same way? Because if that is true, then also in practice there's no reason, no justification of using the PLI over the LC. So I have a very simple data sets of 3 subjects: resting state data, scalp data, MEG. But as you will see, the results are clear cut, so I don't need more data. Yeah, so one thing to realize is that this Gaussian model is not only that you assume that the data is Gaussian, but in fact there are 3 assumptions. So this model is formulated in the frequency domain. So we have complex valued signals, Z1 and Z2. So we can have the real part and the imaginary part. And according to this model, these 2 parts have to be independent. They need to have the same variance, and they have to be normally distributed. So there are 3 assumptions underlying this model. Yeah, so this equal variance assumption is very clear. It's satisfied in all data sets. And what I did is just I take 2 pairs of signal. I take a pair of signal. I use a non-parametric test to test for independence, and I get a p-value. I do that for all signal pairs and for all frequency bands. And then I make a histogram of the p-values. And this histogram is a uniform on 0, 1. And that's exactly what you expect if the null hypothesis is true always. So that's clear. The real and the imaginary part have equal variance. It's in the experimental data, that's clear. Yeah, there seems to be a slight dependence between the real and imaginary parts, in the sense that the histogram of the p-values is not exactly uniform. There's some small deviations. So that's one deviation from the model assumptions, but it's very, very small. Okay, and then the third assumption, Gaussianity. So according to the model, we need that to have the real and imaginary parts to have the same, to be Gaussian distributed. And here I will only show the results for the real part, because those for the imaginary parts are exactly the same. They have exactly the same distributions. And what you tend to see is that these distributions are more peaked than the normal distribution. So here we see the 3 data sets. And the black curves are the distributions of the real part of the Fourier coefficients. Just average over channel pairs. And each black curve is a different frequency band. We have 40 frequency bands from 1 to 40 Hertz. And the green line is the average over frequency bands. So you see in all 3 cases that the green line is more peaked than the red one. And the red one is standard normal distribution. So the distribution of the EEG and MEG data seems to be more peaked. Although the amount of deviation depends on the frequency band. I will come to that. So, okay, this is maybe a bit of a sidestep, but can we maybe find a better model than for these data? And yeah, it turns out that these distributions can be nicely described by the exponential power distribution, which has this form. And this is a generalization of both the normal distribution and the Laplace distribution. Yeah, because if I take beta as 2, then I get the Gaussian distribution. if I take beta as 1, I get the Laplace distribution. And I can also take beta in between 1 and 2. So beta determines the peakiness, the peakness of the distribution. So if you look at these figures here below, so here I have fitted a Gaussian model to the data at 30 Hertz. You see it's a good fit. But here at 10 Hertz, I fitted the Laplace distribution. So that's the exponential distribution with beta as 1. And you see it's a good fit. But here at 5 Hertz, you really need a value of beta that is in between 1 and 2. In this case, it's 1.4. So you can say this is kind of in between the normal and the Laplace distributions. Okay, so if you now look at these deviations as a function of frequency, so here we see 4 panels. The top one, so all of them are organized as a function of frequency on the x-axis. So the first one is just the distributions of the Fourier coefficients, standardized to have variance 1. The second one is the difference with the standard normal distribution. So here you see clear deviations at certain frequencies. See here it's more peaked than the normal one, and here too. And you see that also here in the value of beta. That is this shape parameter of this power distribution. At these locations, it's close to, well, it's closer to 1, so it means more peaky. And in between these points, it's closer to 2, means it's better described by Gaussian distribution. And this lower figure is just the power spectrum. So you see that these deviations happen exactly where you have strong peaks in the power spectrum. So here we have alpha, and here we have beta. So this is MEG, and you see that also in the EG data sets. See exactly the strong deviations from normality you have exactly here where you have these oscillatory resonances in the spectrum. And in fact, this is the data set from Guido actually. It has only a strong alpha, well, and some theta here, some beta also actually, and in fact you can almost infer the power spectrum from these beta values. It really follow each other very closely. Okay, so the conclusion of this is that the deviation from normality are confined to those frequency bands where you have very strong rhythms. This the spontaneous rhythms, right? Theta, alpha, beta rhythms. And in between, it's well described by a Gaussian distribution. Okay, so given that you have these deviations, then the obvious question is, if it's still true that in experimental data, the PLI is equal to the LC, as it is for Gaussian model. And the second question is if the PLI is still a worse estimator for all sample sizes than the lag coherence. So in other words, are these properties of the model, are these robust against these particular violations of the model assumptions? And I think, yeah, I think I will show that they are. But this is what I just said. Okay, let's first look at the sampling properties. So this is now this experimental data. So what I did is I take one pair of electrodes, I compute the phase lag index and the lag coherence. I do that for all electrode pairs and for all frequency bands. I pull everything together and then compute the standard deviation of that. And that's what you see here. On the y-axis and on the x-axis, you have the sample size in number of windows from 1 to the maximal length of the recording. So this recording is the longest, 200 windows. And here you see essentially the same as you see in the model, that the blue line is always higher than the green. Means that for all sample sizes, the lag coherence is a better estimator than the phase lag index. Even though okay, I didn't differentiate between frequency bands. This is pulled everything together, but still, it's pretty clear cut, I think. The second property, so is the phase lag index equal to the lag coherence? It's not so easy to infer from experimental data because you need infinitely long recordings to assess that. So I took the complete observation time. I calculated the coherence, sorry, the lag coherence and the PLI for every channel pair. And then I factorized these matrices. So I have two matrices, sorry, two vectors of length n squared, where n is the number of sensors. And then I computed the spatial correlation between these factorized matrices. And what you expect that if these are indeed the same, these measures, then if the average coherence, sorry, lag coherence or phase lag index is very high, then the spatial correlation would approach 1. So on the x-axis, I plotted the average spatial correlation, just the average of the two vectors. And on the y-axis, I compute the spatial correlation coefficient. And you see indeed that they approach 1 if the average value becomes very high. And yeah, for low values, you don't have that because everything is dominated by sample, by statistical noise. So this is kind of an indirect argument that the PLI and the LC are equal, also in experimental data. For the MEG, they don't go up to 1, but I think it's because the average value doesn't become very high, and the recordings are also a bit shorter, so there's more sampling variability. But I think this is pretty, yeah, it's pretty convincing evidence that these results are robust against the violations of these model assumptions. So I think the conclusions are clear cut. Yeah, the PLI and the LC are estimated of the same quantity, both in the Gaussian model and in experimental data. And the LC is a better estimated than the PLI in the model, but also in experimental data. And the last, which I think so a lot of people couldn't care less about, is that these distributions can be modeled by this exponential power density. That might be interesting for methodologists in the future, because you can construct maybe more general models of these data. Here's some references. Thanks.
Thank you very much, Victor. Just a very fast question. Did
Yeah.
you calculate whether the differences between PLI and LC were significant in these cases?
Significant.
Mm-hmm. Statistically significant.
Well, okay, so if you want to compare the performance of two estimators, you have two aspects. You have the bias, and you have the variance. So the bias is neglectable actually. So I only concentrated on the sample variance. Well, I showed that the LC has a smaller sample variance. So in that sense, it's a better estimator. So I don't need to perform any statistical tests because I just generate from the model itself.
Okay.
I mean, yeah, the conclusion, yeah, it's just more statistically variable than the LC. And that's a separate question than statistical significance. Yeah.
Okay, thank you very much.
Okay, let me stop share.
Okay, very nice presentation.
Yeah. Make them new to. Yeah, there are many things there to say, but if time is up, time is up. So what shall we do? To Richard, yeah, you're right. It's a circular Gaussian. You say them additional, but I assume it's circular Gaussian, not general Gaussian.
Yeah.
So I wanted to say to Sepide, when you compare, are they correlated phase coupling and amplitude coupling, what you study? But it could be that just phase coupling is amplitude coupling is the square of phase coupling. Then you look at a linear relationship when you look at a correlation, but it could be that you see that you get a higher value when you calculate when you square it first. So it's not clear. But I don't think in your case the results will be different, will differ so much. But in principle,
You mean for the amplitude correlations?
Yeah, when you say,
Yeah, I didn't look at that. Yeah.
yeah, what's Sepide? There was Sepide. So when she said, we look, is phase-phase coupling correlated with amplitude coupling? But it could be that it's not correlated, but the square of phase-phase coupling is correlated to amplitude coupling. I don't think so this is relevant, but in principle, you should be aware that the relationship doesn't have to be linear just if they measure the same thing. It can be a square of one is the square of the other, not the same. Not so important, and time is up anyway. So,
well, maybe I can ask you one question, Guido.
sure.
I would be interested in your opinion. So, okay, these amplitudes they really deviate from this Gaussian model.
Yeah.
So have you any ideas about a more appropriate model?
Then a nonlinear. Yeah, that's a good question. For alpha rhythm, you mean make a nonlinear dynamical model which generates alpha, which better reflects it?
Or just a statistical model, yeah. But
a statistical model for the distributions, you mean? But
yeah,
but you had the von Mises distribution. One could also do that. I've seen a paper where they compared it. Circular Gaussian with von Mises for the phase distributions. That was for the phase distributions, but they couldn't say which is better. But I haven't seen what you suggested. Yeah,
yeah, but I mean, like if it's true that the amplitude dynamics is in real brain is kind of independent of the phase dynamics, then you should have models that allow for that, and the Gaussian model does not allow for that.
No,
right?
that's true. Yeah, that's true. So I don't know. I don't have a better model for that. But I want to say, but there's one thing I can confirm that PLI. I also looked at it, not in all detail as you did, but qualitatively also found that PLI is statistically not so good. Just using the sign without amplitude, it ruins it also statistically a little bit. Not too much, but it just is worse statistically.
And maybe I can ask one other question to Alessandra. Or?
Yes, of course.
Yeah, I was wondering. I can imagine that with this two-person setup, it must be very challenging to deal with like a motion artifact, because there might also be coordinated, no, between the persons. Could you say something about that?
Yeah. So. First of all, so this is the reason why we divided our setup and our experimental design in two parts, in which we have on one side the naturalistic interaction and the intake, in which there is literally an open communication between the patient and the clinician, and we cannot even hear that. So we are in a room in which we can see them to be sure to know when they are done, but we cannot even interfere with that. And that's very challenging in terms of motion artifact correction, and we are still working on that, and that's the reason why I still don't have final results in that part. It's literally the main reason, because even though we have a lot of data, so we are in a good spot in terms of for example, literally rejecting the specific part of the trace, right? Because if we have 30 minutes of intake, of course, we can isolate specific windows of interest. But from a behavioral point of view, these windows of interest always have one of the two person at least speaking or moving. And so it's very challenging because you have to reject the same trials and the same epochs in both. And this is the reason why, in order to have more robust results, while we figure out what's the best way to deal with these motions artifact, and there are algorithms we are trying with the good performances. We also have this pain task, which is the task in which we ask them not to communicate verbally. And so we have our data are much cleaner. And for example, in this data set that I presented today, there is a part of ocular links like rejections and motion artifacts, but it's not higher than in a normal experimental design I worked with before.
Okay, thank you.
But yeah, that's a very big issue in hyperscanning, because most of the time if there is a verbal communication, there is a lot of muscle going on.
Yeah.
I can comment on the work by Professor Norihiro Sadato in Japan. The first hyperscanning study he did was from 2 subjects in different cities connected by internet. So it was
really? That's a cool.
Later, he created his lab, and he had 2 MRI and 2 EEG equipment. But in this case, one was in Fukushima, the other was in two different cities just connected by internet.
Well, with the COVID and hospital restrictions, so like we really risked to do that again in the past months. So yeah. And may I ask like a question to Sipide? Okay. So like I'm really interested in this large scale analysis. And so I was curious to know, like just to have a better understanding in terms of the role of different frequency bands. You showed like the correlation, like the coupling between the measures extracted from like neuroelectrical measures and fMRI. And we could see that there was like this correlation was higher in beta band. So I was wondering if that was true in that specific case, because that was acquired during like a specific task, or it was a resting state, or if that's true in general, that there are specific frequency bands that correlate better with the fMRI.
So you're talking about the spatial similarity of the static time averaged connectome.
Yes.
So all of the data that I showed from our lab are concurrently measured. So one issue with concurrent data is that the MRI gradient artifacts affect the higher frequencies worse than the lower frequencies. We have much cleaner data now, but we haven't analyzed it yet with simultaneous multiband fMRI, so much more rapid sampling, denser sampling, and also better cleanup of the data. So I wish I could tell you something about that, but when you look at the traditional gradient template subtraction approaches, so essentially what you do is you generate a template over the many repetitions of the gradient artifact, and then you literally subtract it out of your data. You look at the power spectrum of the EEG before and after, you see that the gamma band and upper beta band is much more lost than the slower frequencies. You're just literally subtracting out some of your signal. In terms of offline data, we haven't compared those directly. So for static time averaged connectomes, you could, of course, do offline. What you need online data for is when you compare the temporal dynamics. I'm thinking back about to the data from HIP and Siegel has done some comparisons between offline MEG and fMRI, as well as several other data, the core beta data that I showed. Trying to think if beta has a stronger similarity of the connectome. I think generally so. We actually wrote a review paper. I would have to go back to it in 2020 in Network Neuroscience. I really summarized most of the studies I knew to that point on the topic. And I seem to remember that the relationship of alpha and beta band to the fMRI was more robust than in the other bands. That might be a property of the spatial scale that we're looking at, because what one would assume that perhaps the gamma band is more tuned towards spatially slightly more local connectivity. However, the dynamic data that I showed at the end, where we're seeing the same very large scale network coactivation patterns in the gamma band as we see in infrasol fMRI, kind of speaks a little bit against that. You really see the same spatial patterns, just at a much more rapid scale. So that was pretty surprising to us in that sense. Because, the implicit assumption under which most of us operate is that what EEG and MEG are imaging is more or less the same neural population that fMRI is able to see. Of course, we see different speeds of their activity of those same neural populations. But it turns out that our data is much more compatible with a notion where the neural population is driving or primarily captured by fMRI versus EEG and the different EEG frequency bands are actually perhaps not very overlapping. And I'd love to continue that discussion, but I don't want to take too much time because it goes a little bit away from your question about the different frequencies. I do think these might be driven by non-overlapping neural populations.
Yeah, it's a very interesting point. Thank you so much.
But in the case of fMRI, you talk about the infra low frequencies from 0 to 0.1. Are we able to see differences in connectivity between in this small range of frequency, or we should consider this as only one frequency?
The problem with fMRI is that even with much more rapid imaging that we have available now, we are subject to the hemodynamic low-pass filter. It doesn't matter how fast we image. All we can see is the neurovascular response to the metabolic demands, right? And those are just slow. The neurovascular coupling is just slow, and we just can't get over that. It seems that that signal is better understood as a center frequency free fluctuation rather than any specific oscillations. We could break it down into frequency bands for analytic reasons, but it's not the same as assuming there are true oscillators as we think of in the canonical oscillation bands. And so there was an early study by Steve Smith's group in the mid-2000s, I believe, where they looked at the different essentially frequency bands of the fMRI and looked at whether they can find these intrinsic connectivity networks in them. And you can see them in the different areas, but it has, the main power, if you're just looking at the power spectrum of fMRI, is really in that extremely slow area. And it seems to be very, very slow fluctuations and conceptually not making much sense to break those further down into frequencies. In fact, I mean, if we did, again, if you have methodological questions, that could make sense, but from a biological standpoint, you wouldn't, I think, get at different neural processes in the different frequencies of the very, very slow end.